Scientists at Lawrence Livermore National Laboratory are developing a forensic technology that uses the proteins found in human hair to identify people and evidence. It will be used in the same way that DNA is used in crime scene analysis.
So one of the things that we got really interested in, and also some of the gaps that existed in the community that we support, which is law enforcement primarily in the forensic community, the broader forensic community, is this reliance on subjective methodology. And was there an alternative or an additional method to DNA that would allow for a very science-based, statistically valid methodology for human identification, but that didn't rely on DNA? Hairs and fibers have been used in forensic comparisons, but that again falls back on some more subjective methodologies where you're making observations rather than technical measurements, scientific measurements. And so what we've done is we've developed a method that utilizes the protein that's contained within the hair shaft itself to give us detailed information about a human, a specific individual, and enable science-based comparisons between pieces of evidence.
Using our current sample sizes, we're able to get enough markers so we can get a unique pattern for an individual that would distinguish them uniquely among a population of 1 million. And what we would like to do is to be able to get that degree of discrimination from a single hair. We're not quite there yet, but we think a combination of improvements in sample processing and also improvements in our instrumentation method.
People sort of light up and they get excited about it because this is a novel approach. It's sort of what we call orthogonal to other established methods, and so it could expand and solve some of the problems that are currently there in the forensic science field. How to deal with mixed DNA samples or forensic samples where many contributors are there. How to deal with hair, which doesn't have a lot of DNA in it except for mitochondrial DNA. How to deal with forensic samples that might be compromised. They've been in the environment too long, the DNA is broken down. So with all of these factors, it's harder and harder to extract the DNA based information. And so what we're doing is another tool, and it just extends, maybe even just a little way, extends what we can actually extract, the information we can extract out of that individual.
So right now we're really in research mode, and so when we do analysis of hairs, it probably takes us a total of 2 days. And so that involves processing the sample, applying it to the instrument, doing the data analysis, and then doing the statistics on the end. We are working on methods to make this much more streamlined. And so once it goes out of the discovery mode in the lab, we're going to work on methods that make it much more routine, so it can now be used routinely in forensic labs that may not have the kind of a state-of-the-art instrument we have, but it can be adapted to instruments that are commonly found in crime labs.
Amazing effort that could only have happened when we have a team here together and all playing and having a different role, and that's one reason why Lawrence Livermore is perhaps the best place for moving this forward and developing this idea.